So in the Google World, remember I put in the student-made review PowerPoints. So there's by chapter if you wanted to go into more detail. Also, I shared this PowerPoint with you, and if you look at the speaker notes, mostly this is just a graphics version of a review, but I put the source for all of the pictures, and those sources a lot of the times are good places to go if you need further research. So like I ended up finding the Bio Ninja. I mean, I watched his videos before and I've always liked his videos, but I didn't realize he had a lot of good content on his website. So if there's any of these topics that when I go over it, you feel like you don't quite know it, maybe click the link that goes with the pictures and see if that doesn't fill in the blanks for you. I'm not going to necessarily reteach. I'm going to point out what I'm looking for on the test. You remember on the review packet I gave you, I highlighted in blue the items that were on the test. And then when I made this review, I compared it to your review sheet and I compared it to the test as I was creating it. And there's a couple items that maybe I highlighted in blue that didn't make the cut. So I'll point those out as we go. This week's schedule, remember today's a Wednesday schedule, and then our exam is on, I see now I need to leave the days on here. Tuesday, Wednesday, Thursday. So our exam is on Thursday at 8:30 a.m. We will be in Illuminate Ed. We won't be in the lockdown browser. Okay. I will have my eyes on you. So we'll meet in the Zoom. So here we go. Here's the test, 61 questions. I'm assuming an 80-minute test according to the time frame that AP Board gives you. So your multiple choice portion of the test, you'll like in May will be 63 questions and 6 grid-ins. I'm giving you 53 multiple choice and 3 grid-ins, and that's your math part. And so like that would be the equivalent of 80 minutes. So we have the 90-minute time frame. You can expect you're going to use the majority of that taking the test. Calculations, you'll have 3 calculations. One, you need to calculate solute potential. You need to calculate water potential, and you need to calculate a chi-square based on a genetics outcome. Okay, here's a link. I'm not going to hit that now, but that's for you. Those of you who would like more work with water and solute potentials. So remember the formulas, and you'll have a formula sheet. Water potential is that psi, which is equal to pressure potential plus solute potential. Solute potential is always negative. Pressure potential in an open container or in animal cells is going to be 0. So you only use pressure potential really when you have a plant cell. So the big thing is you need to be able to calculate solute potential. Remember that's negative I CRT. I being the number of ions it breaks into in solution. You usually will be working with covalently bonded organic molecules, in which case they don't ionize, right? Only ionic molecules will ionize. And I'll tell you on tomorrow's test, or this week's test, you'll be working with like sucrose or glucose. So the ionizing number is going to be 1. Concentration, the molarity of the solute. So you'll have, you know, like a 0.25, 0.15, 0.5. It'll be given to you in the problem. The R is a constant, 0.0831. We used to have to memorize all of this, but now it's given to us. And temperature is in Kelvin. Remember, you need to add 273 to the Celsius degrees. Okay. Chi-square, we've been working with, that's the sum of the observed minus expected. You figure out expected based on a Punnett square, right? Square each of those versions divided by the expected. Observed is what you're actually seeing in the results. You saw 35 purple flowers and 12 white flowers. You expected 75% to be purple, 25% to be white. So calculate that out. What is the whole number that would be? Okay. You will be given the formula sheet. The formula sheet is at the end of the test. And in May, you'll have the formula sheet as well. It's 2 pages. It's a single page, double-sided. And it's good to be able to find where this is at. But these are the only 2 pieces you're going to need. You're going to need the chi-square. So the formula is there. Remember, the degree of freedom is 1 less than your variations. So if you were working with purple, white, and pink flowers, 1 less is 2. If you were working with tall and yellow, tall and green, short and yellow, short and green, your degree of freedom would be 3, right? So 1 minus, and then we only will be working with the 0.05. Notice their table is sideways compared to what you're used to. You're used to degree of freedoms being over here, kind of on the Y, and the p-values to be across the top. So notice it looks different than you're used to. So 0.05 and your degree of freedom, 2, 3, 4. That would be the number. Remember, you want your chi-square to be less than that in order for it to be acceptable, right? If it's higher than that, we fail to accept our hypothesis. All right. You also are given water potential, and it tells you what each of the units are. And you're given solute potential. So you don't even have to memorize how to calculate Kelvin. So all you have to do is know how to use the formulas. Okay. Any questions on the math piece? Okay, so Josh, the formulas are given to you. Big timer in front of you. Okay. So I go through this chapter by chapter. So what I'm putting on here, remember, this is what will be on the test. So your review has a little extra because it's a semester review, but we only have so much time. So first of all, in chapter 1, the only things you're going to really need to do with the scientific method is identify independent and dependent variables, interpret results, and form conclusions. Okay, so you'll be given experiments and it'll ask you, what is the independent, what is the dependent, what would be a conclusion, okay? So remember, the independent variable is the thing that you are personally changing. The scientist wants to see what color light affects growth. The color of light would be the independent variable. The dependent variable is going to change based on the independent variable. If you got green, it didn't grow so much. If you got red, it didn't grow so much. If you got white, it grew a lot. You're going to measure that, right? So this is the data. Chapter 2, so there was questions, do I need to know, actually I think functional groups are in group 3. In group 2, you really just need to know about water's properties and what causes cohesion. So remember, cohesion, molecules of the same kind sticking together. So think of the polarity of water. The hydrogen of one molecule is attracted to the oxygen of a different molecule. Adhesion is what allows it to be a solvent. So it's the universal solvent. So adhesion, the positive portion of hydrogen from water, is attracted to a negative portion of some other molecule like salt. And that is what allows it to pull it apart. Those are the only properties of water you need to be familiar with. Okay? Stop me at any time. Chapter 3, it was asked if you needed to memorize functional groups. There are no functional groups on the test. Carbon and the categories are what you're going to need to know. So characteristics of carbon, like why is this ideal for basing all of our life on it? Because it can make single, double, triple bonds. It can make rings, it can make straight chains, it can branch. So it's very versatile in its bonding, right? And then you do need to know categories, be able to recognize examples and functions. So our categories are the saccharides, the carbohydrates, the nucleic acids, the proteins, and the fatty acids. Fatty acids, I think on the test I asked you about its job, its insulation. Proteins, we are going to need to know a little bit more about because we talked about primary, secondary, tertiary, quaternary structures. Nucleic acids, genetic material, carbohydrates. Recognize that, remember, glucose, cellulose, glycogen, this was on your review sheet, all are polysaccharides, versions of glucose. So you will have questions about the structures. Remember, primary is the amino acid sequence. Secondary is either alpha helix or beta pleats. It's due to local folding hydrogen bonds between nearby amino acids. Tertiary is like whole molecule folding. So you can see after you have a helix, now new pieces are available to make bonds with amino acids that are further down the chain. These are typically because of your hydrophobic and hydrophilic interactions. Quaternary is the whole structure, add a bunch of polypeptides together. Ultimately, this shape determines its job, and if you mess up the shape, you mess up the job, right? We talked about pH and temperature. So this is about, we need to be able to compare prokaryotes and eukaryotes. We need to be able to compare plants and animals. I think the only real structure you need to know what it does is the mitochondria. So this is comparing prokaryotes and eukaryotes. Basically, remember, prokaryotes they don't have a nucleus or a membrane-bound organelles. They do, however, have ribosomes and cytoplasm and genetic material. Plants versus animals, both in function, like during cytokinesis. Remember, we use centrioles for mitosis, and we have the pinching inward during cytokinesis, whereas plants have a cell wall, so you're going to see the cell plate. All right. Other than that, I think mostly just central vacuoles and the cell wall made of cellulose are going to be your big things to know. It's pretty easy. The endosymbiotic theory, remember, the basic idea is this is how compartmentalization came to be, how inner membranes came to be. The engulfing of a photosynthetic bacteria gave rise to the chloroplast. An engulfing of the aerobic bacteria, one that could use the oxygen, gave rise to the mitochondria. A little bit about the cell membrane, and then a lot more about movement. So be familiar with the structure of the cell membrane, specifically that phospholipid piece. The hydrophobic tails are the inner portion. They orientate themselves away from the liquid. The hydrophilic head is the portion that is towards the cytoplasm or towards the extracellular fluid. That's the biggest component of the cell membrane that I'll talk about. So all of the proteins that are involved have many jobs. I think the only job we're really looking at is that has to do with cell transport at this point. So types of diffusion, remember, you have osmosis for water. Facilitated diffusion uses proteins. Carrier proteins move large molecules. Channel proteins move polar molecules or ions. Then comparing to active transport moves against the gradient from like climbing up a hill from low to high rather than high to low. Similarity, they may both use proteins. Co-transport, that would be like your sodium-potassium pump, where the movement of one pretty much powers the movement of the other. So there was a question in yesterday's questioning that you guys gave me about co-transporters. So that's when the two are coupled, the movement of one and the movement of the other. You won't need to know about these other words, just the co-transport. So solutions, remember, okay, hypo, hyper, and iso, you're going to have a number of questions about these. You should be able to recognize the description, like hypertonic has a high amount of solute, hypotonic has a low amount of solute. Be able to recognize the cause and the effect. So like hypertonic is going to cause cells to plasmolysize or shrivel. That's the effect. The cause is that movement of water from its high to its low concentration. So the hypertonic solution attracts water, so water will leave the cell. And the opposite is true of hypotonic. Hypotonic solutions have low solute, so it has a greater water potential. Water will move into the cells, causing the cells to swell, burst, cytallicize. Okay, so be able to predict movements based on, given a situation of how much solute is inside the cell, how much solute is outside the cell, which direction will molecules move. This is similar to a lab we did where we had to figure out the molarity of a potato. So you'll have a graph and you'll have to be able to identify the solutions or the potato, for example's molarity. Remember the point where it crosses 0, it is isotonic. It's neither gaining nor losing mass. So at this point, you can say the plant cell would have the same solute concentration. So this is a little more than 0.1, right? If, remember this is the change in mass. If it's losing mass, if the number is down below here, water's coming out of the cell, which means these are hypertonic. So anything above 0.1 was hypertonic to the cell. No, I lied. Anything below 0.1, because it was losing mass, if I continued my trend line, was hypertonic. If it's gaining mass, water's going into the cells, so it's sitting in a hypotonic solution. So if it's gaining mass, anything above 0.1 is hypotonic. Okay. Remember endergonic and exergonic. It's been a long time since we've looked at those. Ender, taking in, exiting, losing, right? And it has to do with energy. So here, my reactants, what I'm starting with, the products have greater energy than the reactants. So they took in energy, so that's endergonic. In this case, the products have less, so the reactants lost energy. That's exergonic. You're not going to need to know about spontaneous and the change in delta G. Just recognize which is which from a graph. Okay? So ATP, remember, it breaks the bond between the second and third phosphate because it can easily transfer energy from this molecule to other molecules or reactions. It is the most commonly used energy source in cells. Lots of questions on enzymes. So if you need a review, hit the review. I would say this is like, if you did percentages, this probably has more questions than any other topic because they're involved in every reaction, right? So, so how do they control the rate of chemical reactions? Remember, they're lowering the activation energy. They lower the activation energy by putting molecules in proper orientation for reactions. The active site where they're bonding If I had another site, the allosteric site, that comes up in a minute. So the substrate, the active site, and the enzyme, just some general vocabulary. Coenzymes, like coenzyme A, for example. NAD, FAD, for example. The coenzyme, so here the active site doesn't quite fit the substrate. You need some help to fill in the gap. That's what a coenzyme does. It helps the substrate better fit the enzyme. Allosteric is the other site, right? So you have the active site and the allosteric site. This allosteric site may change the enzyme into a shape that no longer fits the substrate. So that would be inhibition. Sometimes it changes it into a shape that fits, in which case it's an activator, but that's not on the test. Just inhibitions. Competitive inhibitors are when there's two molecules that look very close to the same. And if you want molecule A to be broken down, but molecule B is sitting in the active sites, that would be competitive inhibition. They can't work on A. Feedback inhibition, when what you're creating goes back and acts as an inhibitor on any one of the enzymes. So usually this is synthesis reactions. You've made plenty of D. You don't need to make any more D, so stop the assembly line. So it'll stop one of these enzymes, either acting competitively or allosterically. We talked about things that can affect enzymes' activities. So denaturing, usually that's when the temperatures are too high, and that's going to affect with hydrogen bonding. Or when the pH is too low, too acidic, and again, that acts on those hydrogen bonds. So be able to interpret one of these graphs and tell me why it's climbing or why it's lowering. So in the pH, we know this all has to do with hydrogen bonds and shapes. With temperature, this first portion doesn't have to do with the shape at all. This first portion has to do with kinetic energy. Molecules are moving more quickly, and therefore they are more likely to collide, bond, and have reactions. Okay? Above a certain temperature, that's when you talk about denaturation. When you talk about concentrations, the greater the concentration, the greater the rate until you've reached a point of saturation. So at that point, all enzymes are used. You can't go any faster than that. Chapter 7, chemiosmosis is the biggest piece. There's I don't think there's really any questions that directly relate to photosynthesis, so don't spend a lot of time there. What you want to be familiar with from photosynthesis is the electron transport chain, and it's also in cellular respiration. So this cytochrome C molecule here and here, notice this is cell respiration, ETC, this is photosynthesis, ETC. The same molecule plays a role in both electron transports. So it's playing a role in the pumping of the hydrogen ions against their gradient. Anytime you say pump, you're going against the gradient to create a high concentration gradient that then will diffuse through ATP synthase to make ATP. It doesn't matter which process I'm talking about. These are both creating enzymes or creating ATP with the help of ATP synthase and the movement of hydrogen ions. Okay, chemiosmosis. You do need to be able to discuss that process. Aerobic versus anaerobic. You want to know which reactions are aerobic and anaerobic and what causes it. So basically aerobic being with oxygen and anaerobic without, right? Fermentations are both anaerobic. So is glycolysis anaerobic. Glycolysis happens in the cytoplasm, does not need oxygen. Fermentation happens in the cytoplasm, does not need oxygen. The Krebs cycle and the electron transport chain happen in the mitochondria. They need oxygen. Okay, glycolysis precedes both, so it's part of the fermentation as well as the aerobic respiration. Pyruvate is a molecule that is used by both fermentation and aerobic respiration. You also need to know how many ATP are made at each step. Glycolysis makes 2. Krebs cycle makes 2. Electron transport makes 32. Long story short, the majority of ATP is made during electron transport. We also need to talk about NAD and FAD. These are both coenzymes that help move electrons from one reaction to another. So they're being made during glycolysis and Krebs cycle. They're going into the electron transport chain. Notice NADH is going in here. It will power 1, 2, 3 pumps. FADH2 goes in here. It only powers 1, 2 pumps. Thus, NADH goes in at a higher energy level because it's losing energy all along the way. FADH2 goes in at a lower energy level. This is like stair steps. Less energy, all moving towards oxygen. So they're electronegative and they have an affinity for oxygen. That's why oxygen is needed in order to attract the electrons through this chain. The final electron acceptor, role of oxygen, the final electron acceptor is oxygen, and you will make water. Chapter 9, cell division. Controls of the cell cycle. You're thinking right away about CDKs, cyclin cycles, right? The CDK kind of remains constant, but don't forget about external cues. We also use growth factors. We also use like hormones, growth factors, activators, inhibitors, like the external boundaries, right? So there's chemical signals either inside the cytoplasm or outside of the cell itself that also initiate that reproduction of the cell. You don't need to tell me about all of the phases independently, just the general idea. You're going through a growth phase, you're replicating DNA, you're making material needed for division of the nucleus, the chromosomes align at the equator, and the cell divides. A generalization, okay? Calculate relative time spent in each. Remember this lab. So looking at this, the majority of the cells were in interphase, which means it spends the majority of its time in the interphase. It spent the least amount of time in metaphase and anaphase. So you don't actually have to calculate it out. Relative time based on how many cells are in each phase or based on percentage. I can look at this and say the majority of the time is spent in interphase. The next longest phase is prophase. The next that had the most cells in that phase was prophase, therefore it's the next longest phase. Okay. Cancer versus normal cells. Just cancer cells, remember, they spend less time in interphase. I could go through all these other things about it, but the biggest piece, what you're going to see on the test, has to do with the timing. So they go through their cell phases much more quickly and spend less time in the interphase, which is really where your quality control is, right? And that's why they're all messed up. Chapter 10, the polar body. This was one of the questions in yesterday's feed. The polar body makes it so that the ovum that does survive has the greatest amount of cytoplasm, nutrients, increases its chance for survival, basically, right? The polar body, an uneven division of the cytoplasm. First one dies off. Again, uneven division of the cytoplasm. Second one dies off, which means the ovum that survives has the most cytoplasm. When these two come together, a sperm and an egg come together, remember they bring you back to a diploid number. If both these cells had 23, the end result would be 46. If there was a non-disjunction, one of these cells could have too many or not enough. If it has too many, instead of 46, you might have 47. If it has not enough, instead of 46, you'd have 45. So be able to talk about how non-disjunction would affect the zygote number. Remember, non-disjunction can occur during prophase 1 or prophase, during anaphase 1 or anaphase 2. So that was something we had forgotten last time we talked. During anaphase 1, failure of homologs to separate. During anaphase 2, failure of sisters to separate. So if you were fertilized by a cell that didn't have enough chromosomes, my zygote will have a lower number than 46. If you were fertilized by a cell that had too many, you would have more than 46. Okay. Getting into the genetics piece, gene mapping. So you don't have to figure out percentages or anything like that, but given the percentages, you need to be able to put the correct order. So it's a multiple choice, so you don't have to draw it out, but you would need to be able to say the proper order would be A, B, and C. Find the two that are furthest apart, make those your bookends, and then position whatever are in between based on their reference points to A or C. The product rule and the sum rule. The product rule, multiplication, when you need two things to occur together. The probability of the independent events multiplied together gives you the probability of the overall event. Okay, so in this case, if you needed little a, you have a 1 in 2 chance of this little a and a 1 in 2 chance of this little a. So you multiply them together. The sum rule is when you can say or. The probability of either event occurring. You don't need them both. You need one or the other. Then you add. So if you wanted heterozygous, you could get big A and little a. That's this probability. Or you could get little a and big A. That would be this probability. So you would add them together. That's a multiple choice question. Any questions on that? I know that was one of the ones that came in yesterday's feed. Interpreting a cross. Oh, a test cross. Remember a test cross, you start with one with a dominant trait. You have to breed it with a recessive trait in order to find out if the dominant is homozygous or heterozygous. If all the offspring show the dominant trait, you know the parent was dominant. If some are dominant and some are recessive, you know the parent was heterozygous. Okay, linked versus unlinked. Unlinked genes will sort independently. So you get tall, green, tall, yellow, short, green, short, yellow. If they're linked, they stay together. So you'll only get tall and green, you'll only get short and yellow, or whatever the parent cross was to begin with. So looking at results, you should be able to identify whether the trait was linked or unlinked. So do they come in all different variations or do they stick together? So a dihybrid cross, remember to use the FOIL method. The first from both pairs, big R, little r, oops, within one parent. Big R, big Y, outer, the outermost, big R, big Y, inner, the two inners, big R, big Y, and last of each pair, big R, big Y. So that one wasn't very difficult. I gave you a very simple one to do here. On the test, it's giving you the results. and you're kind of working backwards to determine what are the possible parent genotypes. You saw this in the practice quiz I gave you, I believe. The multiple alleles, we work with blood types. They're also codominant. Remember, codominant means two exist together. So if you get A and B, both A and B will show. In this one, you have a back cross. So you're giving the parent, the offspring, and the parent blood types, but not genotypes. And you're working backwards to determine the parent genotypes. Here's a couple of examples. This would be a dihybrid cross. I believe this is from the packet I gave you over the winter break. So given a dihybrid cross, these would be the results. So on the test, you're going to be given the results, and you have to determine the parent genotypes. So like if you only get one trait, you know the parent was homozygous. If you get variations, you know they were heterozygous. Again, what you're going to see on the test, you're given the parent's blood types, but not genotypes. You're given the child's blood type, and you have to work backwards to determine the parent's genotypes. So fill in what you know, and then work backwards in the Punnett square. Okay, you guys have that. You can read it later so that I don't take up too much of your time. There was a request to go over sex-linked traits. You don't have any Punnett squaring with sex-linked traits. You only have a pedigree that has to do with sex-linked traits. So for the sake of time, I'm not going to go through how to do the Punnett square. You do need to be able to identify what the inheritance pattern is and who a carrier is. So remember, if it shows in every trait, it's dominant. If it shows up equally between two genders, it's autosomal. If it seems to favor a gender, it's sex-linked. So like sex-linked dominant usually shows up in females. The only carriers are when you have a recessive trait. So again, if it skips a generation, it's recessive. If it appears equally in both genders, it's autosomal. If it appears to favor the males, for example, it's sex-linked. In this case, you don't have a skipped generation, except you don't have a skipped generation. So that's not always the case. Recessive, those are the ones that are going to have carriers. So in this case, autosomal, you have to get it from both parents. So both 1 and 2 would be carriers. In this case, these boys got the X from their mom. Mom shows, so she's not a carrier. Mom got it from her dad, and she would have had to have gotten it from mom as well if it was recessive, right? She needs 2. The male only needs 1. That's why it's more common. The female needs 2. So this mom here, number 1, would have been a carrier. So given a Punnett square, identify the inheritance pattern. Given a Punnett square, identify who's the carrier. Only true and recessive traits. So look for the person who doesn't show, that gave it to their child. That takes you through all 61 questions.